Hello, I'd like to welcome everyone. For those that I didn't meet yet, I'm Megan Jindrysik. I'm the co-chair of the Boston Seminar Series and the one who sends all the emails. So I know you've all seen my name. So welcome. Tonight's very exciting. We're going to have our speaker is Earl Miller, and Nacho will give you more of an introduction of him. I just wanted to let you know about a couple of other events that we have coming up. On May 1st, we'll be presenting another event, Fireside Chat with Marty Culpepper and Whitney Espick, and that will be here on May 1st at 6 o'clock? Yeah. 6 o'clock, 6:30 again here. There'll be more information about this will be on our website very soon, this week. Also, May 7th is the MIT Club of Boston's annual meeting. And that will be held at the Broad Institute at 6:15, and all are welcome to come to that. And please do and find out what's going on at the club, what we have planned for upcoming events. The speaker will be the MIT Alumni Association president, Una Park. Will share her insights with us then. Again, you can find also more information about that on our website. And we kind of take a hiatus for the summer with the Boston Seminar Series, but we're working on putting together an exciting program starting next fall. And if you enjoyed learning about exoplanets, we tentatively have scheduled Professor Sarah Seeger, who worked on TESS, to be one of our speakers. So please keep a watch on our website and read our newsletters, and we'll have more information about dates and other speakers coming up.
Thank you, Megan. After this nice session, I think I have to read today because that's going to be too difficult for me. Working memory to zero. So working memory is the most fundamental cognitive function. It is the sketchpad of consciousness, the arena where you do your thinking. And for the past 50 years, working memory has been thought to rely on cortical neurons that fire continuous impulses, that keep thoughts online. Okay, that's something. We're going to talk about this, okay, I think we're done. Okay, just a couple of more sentences, yeah. However, new work from Miller's lab has revealed more complex dynamics. The impulses fire sparsely and interact with brain waves of different frequencies. In these rhythmic interactions that allow your brain to control the information held in working memory. In other words, a rhythmic dance between brain waves may underlie your ability to control your own thoughts. And then in the same presentation, we had also Earl's bio. You can tell that there's all the awards and everything that you can get here. So it's great to have Earl's tonight. Just one final comment, and if you allow me this, because we discovered recently that Earl has among his interest and curiosity, which is this science. He loves music. He's passionate of music, and I didn't know that some of those scientists, they use their neuroscience conferences to practice with other scientists. In that case, he's in a rock band that maybe you want to talk eventually, but that's going to be something different.
I wasn't planning on it.
It was not planned. Okay. So it's very exciting to really have Earl tonight, and please welcome to thanks Earl tonight.
Thank you very much. Thank you very much for the invitation. It's a really great pleasure to be here. It's always a great pleasure to talk to fellow MIT people. I'm going to stand down here if that's okay. Can everybody hear me? Great. So let me tell you a little bit about what I do. I'm a neuroscientist. I've been a professor of neuroscience at MIT for nearly 25 years now. I've had many opportunities to leave MIT, but no one could lure me away because I love MIT. And who doesn't love MIT, really? So here's what I do. What I do is we try to reverse engineer the working of the brain by recording electrical activity for the brains of animals performing tests. That's our main method. We also use fMRI and measures of electrical activity in humans, but really getting down into the brains of animals is the main way. We get down to the nuts and bolts of how the brain actually does stuff. And what we measure, I'm talking about two different measures today of brain activity. One is spikes. Spikes is the jargon that we use as neuroscientists to refer to pulses from individual neurons. All the millions and millions and billions of neurons in your brain, they all give off these pulses which we call spikes. And then I'll be talking about something called local field potentials, or LFPs. LFPs are the mass action of millions of neurons near each recording electrode. It's just the summed activity of a whole bunch of millions of neurons all firing together. If you guys are all familiar with EEG measures, if you stick a bunch of electrodes on your scalp, you get brain waves. That's like LFPs, it's only outside the skull. If you get down inside the skull, you get a more precise measure, but it's essentially the same kind of measure. You're measuring the activity of lots of neurons acting together. So, today's topic is working memory. So, why should you care about working memory? You should care about it because working memory is what you're using right now. Working memory is a sketch pad of your consciousness. Working memory is what you use when you think. It's what you use when you plan. It's what you use when you deliberate. It's what you do when you weigh different decisions. It's really where all the critical, conscious thinking of your mind takes place. It operates over a few seconds, so it's the current contents of your consciousness. For that reason, it's temporary. It manipulates information when you're thinking about what to do, when you're planning your day, when you're thinking about juggling around thoughts in your head, you're manipulating them. It all takes place in this thing called working memory. And it also involves focusing your attention on relevant things. So right now, you're using your working memory to realize that there's a speaker at the front of the room, and you should be paying attention to the speaker, hopefully, not to make any assumptions. Now, part of the brain, this is a product of many parts of the cortex of your brain, but the main part is this part called the prefrontal cortex, also known as the brain's executive. Now, what do I mean by brain's executive? It's the part of the brain that really keeps in mind what goals you have and makes you behave appropriately in any situation you're in. Now, one of the ways we know, there's many ways we know that the prefrontal cortex acts as the brain's executive. By the way, if anything I'm saying here, feel free to stop me and ask for further clarification. I'm happy to take questions anytime. So one way we know the prefrontal cortex is the brain's executive is because of this unfortunate character named Phineas Gage. Phineas Gage was a railroad worker who in 1849 had this metal rod blown through his head as a result of an accident. And you see the skull here, it entered through the bottom of his cheek and entered the top of his head, destroying much of his prefrontal cortex. And what happened, he could see fine, his senses were unimpaired, his memory was fine, his motor skills were fine, but what changed is something very high level. His personality changed. He went from a sober, responsible, nice human being to somebody who was impulsive, profane, inappropriate. And what basically happened is his thoughts and behaviors were governed by reflexes, his reflexive reactions to whatever was outside in the environment. There was no filter in his brain anymore. If something occurred to his, some thought popped up into his head, he said it, or he did it. He was walking to work in the morning, he walked past the local pub. There used to be a tradition in this country of having morning beers for workers on the way to work. He'd be walking to work after recovering from the accident. He'd walk into the pub. As he walked past the pub, and never leave the pub. So basically, his brain was an unfiltered reaction to everything around him. Any stimulus that was out there, any input to his brain, he reacted to without any filtering at all. And as a result, you can imagine he became very profane, lost his friends, lost his job, spent the rest of his life basically as a touring attraction. He went around the world showing off this rod that went through his head. So that's how we know that Phineas Gage, how we know the prefrontal cortex. One of the reasons we know the prefrontal cortex plays this executive role in brain function is the filter that tailors your thoughts, that focuses your thoughts on the things that are appropriate to navigate any situation. And today what I'm going to tell you about is a new model of working memory, because the platform where all this executive stuff happens in the brain is working memory. It's the sketchpad of consciousness where you do all these things that allows you to engage in appropriate actions, to think about goals, to make plans. It's the infrastructure. It's the base by which you make all your important thoughts and actions. So I'll begin by telling you what the old model of working memory was. Actually, it's not quite the old model yet, because it's all, I'm talking about all brand new stuff. But trust me, one day it will be the old model. And the question is, how do you hold thoughts in working memory? So the most basic function, there's this executive part of working memory where you have to think about things, you decide what to think about, you manipulate your thoughts, but the basic function is you're holding thoughts in mind. So how does that happen? Well, the classic model for the past 50 years or so are sustained spiking of neurons. Remember, these spiking are these pulses of neurons are giving off. And here's an example of this. So the way we test working memory in the laboratory is a number of different ways. Typically, we've taken an animal or a human, and we show them a picture or something on a computer screen. We usually use vision, because vision is very easy to test. And we have them remember that for a few seconds. Hold it in mind in working memory. So here's a typical test, or a classic test, where you have an animal fixate the spot, keep their eye on the spot of light. It's called fixation. You flash a cue somewhere in the periphery. The animal's trained not to move their eye. You have to wait over a little delay, and then after the delay, they're given another cue, and the cue tells them now make an eye movement or make a hand movement or point to where the cue was that you just saw a few seconds ago. And that demonstrates we're able to hold this cue in mind for a few seconds. Now what you see in the bottom here is an example of an individual trial, individual one-time running of this sequence of events. And what's on the record here are event codes of spikes happening from a single neuron in the prefrontal cortex. They're kind of piled up here because we're actually binning these spikes over like a 5 millisecond window. So if a few spikes occur in 5 milliseconds, they get piled up. But you can see what happens is that you present the cue, and this neuron begins to fire off spikes saying, I recognize the cue. And then over this memory delay, it just keeps spiking away as if it's holding this cue in memory until the animal makes its response, and then the neuron stops spiking. And if you look over here, this is the, this is one individual trial, one instance of the animal performing this task. Now we're looking at the average across a number of trials, like 10 or 20 trials, so we can really begin to pull out this profile of activity. And what happens is the neuron starts at a baseline firing rate, you present this cue stimulus, the neuron starts firing to this cue stimulus, it maintains some level of spiking over this delay, seemingly holding that cue in working memory, and then when the animal makes its response, the neuron returns back to baseline because now the task is over and the animal can forget it was just holding working memory. Okay? Now, the model of this is that this is how working memory works, is that you have a neural ensemble. An ensemble is nothing but a network of neurons that represents a given thought, or memory, or perception. It's the basic unit that represents information in your brain. And the idea of this classic model is that there's a neural ensemble that gets activated by this cue, and then it just simply remains in that active state over a few seconds while the animal's holding this cue in working memory. And this model is very appealing for its straightforwardness. I mean, if activation of an ensemble is what causes you to have a perception or a thought or a memory, all you need to do is just keep it in this active state where it's spiking along, and you're holding that thing in working memory. But as I think you'll hear by the end of tonight, is that things are never as simple as they seem. The brain is a very complex organ. One of the most complex things we know of, it's hard to follow that satellite launch. The brain is the most complex thing that we know of, and whenever you think you have an answer to something, it always turns to be more complex than you thought. So, the problems with the classic model is that much of our field for many years, for half a century now, has been focused on individual neurons. Now, and the problem is the brain is not individual parts. It's networks of neurons all interacting together in a very fluid, dynamic way. And you can't see those network properties one neuron at a time. Like they ask, how do we get to this state where we've been so focused on just studying the brain, one brain cell? I mean, there's billions of neurons in the brain. How do we get to the point where we're focusing on one neuron at a time? Well, let's go way back to 1962, back when people were trying to first figure out how the brain codes information. And there was a lot of frustration because people were the way that people started to look at this was in vision, because again, vision is very easy to test. And they were looking at neurons in the primary visual cortex, which is here at the back of your brain. Primary visual cortex of your brain, part of the brain that first processes vision, part of the cortex that first processes vision. And they were trying to find a way to activate these neurons, to make sense of these neurons. Well, along came Hugo and Wiesel, who won the Nobel Prize for this work, And at Harvard Medical School in 1962, they were doing these experiments trying to activate these neurons in the visual cortex. And they were having no luck whatsoever. They were using diffuse flashes of light. They were using all sorts of things that were not very specific. Mainly just trying to like flash lights in his eye. And one day, either Huber or Weasel, I forget which, was putting a slide into the projector and the slide got stuck. And there was a dark edge across the screen. And all of a sudden the neuron they were studying started firing. That's what it sounds like when you listen to an audio. It was like firing like crazy. And they thought, and serendipity favors the prepared mind, they thought, we got something here. And what they eventually realized is that different neurons in your visual cortex all respond to edges in particular locations on your retina and in particular orientations. In other words, your primary visual cortex breaks down your visual scene to a bunch of little line segments all over your visual field, different orientations, and that's how you eventually get what you're seeing in front of you. Well, that collectively blew the mind of our field. People said, this is the way forward. They found, for the first time, they found that how neurons code information, this is the way we should do it. We should focus on each neuron at a time, and eventually we figure out the brain. It's as if the brain is a bunch of gears in a giant clockwork. If we figure out each individual gear, we'll eventually figure out the whole brain. Now, I'm here to say that's a simplistic way of looking at the brain, and it's really not the way the brain works. The brain is more interactive, dynamic. There's emergent properties you can't tell by looking at individual gears. But having said that, that work that Huber and Wiesel did was groundbreaking because we can't get to where we are now, and I'm talking about tonight, if it weren't for what they did back in 1962 and beyond. They had to figure out the first approximation of how the brain works in order for us to then figure out the more complex properties. But science also is forward-looking, it's not backward-looking. And at this point, now that we're in 2018, we realize the brain is not as simple as individual neurons all doing individual things. Now, one of the emergent properties that you can't tell, you can't detect from looking at single neurons is something called oscillations or brainwaves. All you guys know about brainwaves, right? I mean, if you stick EEG electrodes in your head, you get squiggly lines, and those squiggly lines ever go flat, boy, you're in trouble because you're probably dead, or you're in a coma, or you're in deep anesthesia. Hopefully 2 or 3 of those things, or one of the 3 of those things. These oscillations, these brainwaves reflect your brain working. And that was actually part of the problem for many years because for like half a century when people were focused on individual neurons, they dismissed this idea of brainwaves. Even though the study of brainwaves predated, we first studied brainwaves 100 years ago. But people, once this single neuron approach took over, people started ignoring brainwaves, and they literally thought of it as like the humming of a car engine. Like if you listen to the humming of a car engine will hum at a quiet level or an intense level, and it reflects the engine running, but the humming has nothing to do with how the engine runs, right? Well, that was what people used to dismiss the idea of oscillations in the brain, is that it's like the humming of a car engine. Now, you guys being MIT alumni, I need to tell you that that's kind of a little bit silly. I mean, again, it was a different, we were operating on a different model like 40 or 30 years ago. But if the brain is a bunch of electrical potentials all interacting, well, having a brain oscillate to different levels of potentials is obviously going to have an effect on the electrical activity of the brain. And we now come to realize that these brain waves actually play a critical role in brain function. And I'm going to tell you one of those roles in a few minutes. Now, another problem with the classic model is this appearance of sustained activity that you show the animal a cue, or a human a cue, and its neurons fire over this memory delay while the animal or human is holding something in working memory. The idea that this is a sustained, ongoing level of activity is largely an artifact. Because when you focus on individual neurons in the brain, the problem is that individual neurons are only one tiny voice in a huge, huge chorus. So the signal of individual neurons is relatively weak. So what people have done for the past 50 or 60 years is average across multiple trials, multiple instances of the same thing. So I may have the animal hold one picture in memory and a second picture in memory, and I'll have the animal hold one picture in working memory 20 times, and the other picture 20 times. I'll average across those 20 instances to get an average picture like this. And the idea, so you're following me in this, so in order to boost signal and decrease noise, the idea was we average across multiple instances of the same event, and therefore we're pulling out a signal like this, and we're getting rid of all the noise. Well, think about that for a moment. Every time you do the same thing, are you always the same person? Are you always thinking the same thoughts? No, your brain is constantly changing and evolving. So what if this noise isn't noise, but brain dynamics operating in real time, because you're not the same person, not doing the same thing every time you do something. There's always going to be a little bit of differences going on. Now, the other part of the classic model is sustained spiking is metabolically expensive. Every time your neuron fires a spike, it costs a lot of energy. So the idea that the brain would evolve to have a memory system that you're constantly using and have neurons in your brain constantly firing these very metabolically expensive spikes is a little problematic. And by the way, when I'm talking about this model, I studied under this current model. I studied this current model, the classic model. I studied it for 20 years before I had my enlightenment, shall we say. So I bought into this model, but this is one thing that always bothered me about the model, is that it costs so much energy to fire these spikes. Why would your brain constantly be firing like constantly? Now, you know, evolution is not intelligent design. Evolution just, things evolve and things happen, and eventually we get to the kind of creatures we are. But one thing that evolution always does is tries to conserve energy at all costs. So it never made sense to me to have a system where a system you use all the time, where the neurons are constantly firing away for these expensive spikes. Now, the other problem is that this model of sustained spiking doesn't actually work very well. So I've talked about experimental observations of how working memory works with a sustained spiking in the brain. Well, what people can do with that is do computational models, computer models with simulated neurons, simulated brain cells, and figure out, well, how does this actually work? And if you do that, what you find in one of the models that's used to, the main model used to model the sustained activity is something called an attractor dynamic model. And all attractor dynamics are saying is that there's stable patterns of activity. So remember I mentioned an ensemble, a given pattern of neurons in your brain, a given network of neurons will represent one thought versus another, one perception versus a memory. And the idea of these attractor dynamic models is that each of these different ensembles, these different memories, are a different configuration of neurons being activated, and they remain stable in the stable, sustained state when they fall into a low energy point that they can't get out of. And if they're in that low energy point, a stable point of activation, then they stay there until something comes along and perturbs it. But that's the whole problem. Perturbing is the whole problem with this model. Is that if you, with these attractor dynamic states, if you add a second item, you say you want to hold two thoughts in mind simultaneously, or two stimuli, or two pictures, or two something, these models of attractor dynamics cannot do it. Because what happens is you try to activate two of these states here, two of these different configurations, two of these states, and the energy lowers between them, and they smush together, they interfere with one another. So computationally, it's been shown that these sustained spiking doesn't work very well because the different things you're trying to hold in mind smush together. Now, I'm going to qualify that by saying we actually have a very limited capacity to hold multiple things in mind simultaneously. The average adult human can only hold about 4 things in mind simultaneously. Keep that in mind, I'll be talking about it later on. So our capacity to hold multiple things is very limited. However, we can hold more than one thing in mind simultaneously, and these models can't do more than one thing. So that's a real problem. The other problem is that any new input to these models, any new sensory input, will knock the model out of its attractor state and completely disrupt the memory. So you and I are trying to, we may be using our working memory to think a certain train of thoughts, and something may be going on in the background, some new noise, somebody else is talking. We can defend our memories against those new sensory inputs. Hopefully we can. You guys are all MIT graduates, so I'm sure you can. But in these attractor dynamic models, it just doesn't work. Unless you pose other mechanisms, other unknown mechanisms, any new input to the system disrupts this stable state and knocks away the memory. So, what have we done? We took a re-examination of working memory using new techniques. What we did is our lab innovated techniques. So back when I started the lab 25 years ago, this single neuron approach was full swing. And I thought to myself, why are we studying the brain one neuron at a time when the brain is networked? We should be studying networks. So our laboratory innovated techniques to record from hundreds of neurons, even thousands of neurons simultaneously, using electrode arrays that we developed. At first, we actually got a lot of flack for this. People would say to us, how can you do careful science if you're studying more than one neuron at a time? Which never made sense to me. I mean, we're collecting this data and analyzing it all post hoc. I mean, what's the problem with collecting more data? Since when is more data bad? But we persevered. I don't want to make myself sound like but we did suffer a little bit of slings and arrows. But now this new approach of studying networks of neurons with arrays of electrodes has now become standard in the field. And so what we did is we re-examined working memory using this new approach, and we examined both spikes and LFPs. Remember, the LFPs are the mass action of millions of neurons near each electrode. And because we were collecting so much data simultaneously, instead of just one neuron at a time, we're collecting data from thousands of neurons at a time, even millions of neurons if you go on the LFP level. We could examine what's happening during working memory in real time without averaging across trials. We could pull out the actual dynamics of what the brain is doing without having to throw away a lot of the details. Okay? So here's what happens in real time, actually, as the animals, because we study rhesus monkeys in this particular experiment. And here's what actually happens in real time as you're holding things in working memory. So this is a plot showing, now we're looking at the LFPs now, again, the local field potentials that measure the activity of millions of neurons simultaneously. We're looking at the frequency of these LFPs, these brain waves. We're looking at on the y-axis. And here's time on the x-axis. The color is the power, so blue is no power or low power, red is higher power. And this is time in the trial. So S1 and S2, in this particular experiment, we're cueing the animal to hold two stimuli in memory, two pictures in memory, S1 and S2. And what happens? What we don't see is anything consistent with the classic model. We don't see neurons beginning to spike and then simply maintain their spiking over the whole memory delay here. In fact, we can replicate that result of the sustained spiking by averaging across many, many trials using these data. When we look at individual trials, this is what we see. What we see are these are bursts of gamma in the LFPs. That's up here. So we have these two different ranges of frequencies. Gamma is above 40 hertz. Beta is 25 to 35 hertz. And what we see while the animal's holding these two stimuli working memory, we see these bursts of high frequency gamma oscillations. And associated, riding on top of these gamma bursts, are the spikes that are carrying the information about the two stimuli the monkey's holding in working memory. Okay? So you see that here, here, here. And down here are beta bursts occurring at a lower frequency band, more on those later. But the information, the stimuli, the cues, the pictures the animal's holding in working memory is being carried by these high frequency gamma bursts and the spiking associated with it. So you guys are with me? Groovy. So, but this immediately raises the question of, well, what is actually maintaining the memories? If all this spiking taking place, these gamma bursts are taking place very sparingly, not in a sustained way, and there are these gaps of time between them, what's actually holding the memory between these events? Well, we found evidence that the memories are being held by impressions left in the network. So these neurons are all connected into a network, and they have connections that can be modified. They can be strengthened, they can be weakened. And we found evidence that every time there's a gamma burst with the spiking, what happens is it temporarily changes the weights, the connections between the neurons, leaving an impression in the network that lasts for just a half second or so, or second or so between these episodes of gamma bursts. So it isn't that the working memories are being held by this sustained state of activity, it's being held by this very sparse activity. It's happening occasionally, with lots of rest in between, and in between, the memories are actually being held not by the sustained state of spiking, but by these impressions that the spiking is leaving in the network. Now that sounds like a trivial, maybe energy-saving mechanism, because now you don't have to spike as much, because you have all these gaps in time where you're not wasting energy on spiking, but it actually has profound implications for how working memory works. One thing it does, allows multiple thoughts to be held in mind without interference, how to work without interference. Now, if you assume, if this is true, we have these very sparse events here, and the memory is being held by impressions in the network between the events, now you can hold multiple things in mind. Because the problem with the sustained activity of tracker dynamics models is that the sustained activity is very labile, it's very easy to disrupt. Well, now you only have this active state, this vulnerable active state happening very rarely, which means you can hold multiple thoughts in mind simultaneously. In fact, that's what we found, is that these different gamma episodes, these different episodes here, are different memories being activated. So essentially what the brain is doing is, over time, juggling the different memories you're holding in mind simultaneously. If you're holding 2 or 3 things in mind simultaneously, your brain is juggling them by activating them at different times so they don't interfere with one another. So that solves that problem with the classic model of sustained activity. The other thing it does, it allows working memory to be more resistant to distraction. Remember, I mentioned that we're all very good, especially MIT graduates, are very good at focusing their thoughts and ignoring distractions, keeping your mind focused on a goal. The classic sustained activity and tracker dynamic working memory model is very bad at that, because any new input just disrupts the sustained activity. But now we're only seeing very brief bursts of activity. That means that any time that there's no activity going on, a new distracting input could come in and it has no effect on the system at all. So all the system has to do is protect the working memories from these brief moments of activation. So now we see these two improvements, 2 big improvements over the classic model working memory, solve 2 major problems of the classic model. So in other words, these 2 things alone solves many of the problems of the classic model. But what it also solves is something that's never been addressed by the classic model of working memory. One of the most important things, the most important thing about working memory, is that it's under our control. We have volitional control of our own thoughts. Think about what your inner life would be if you didn't have control of your own thoughts. All your thoughts, all your actions would be buffeted by the environment. Whatever thing comes along, you'd be like Phidias Gage. Whatever thing that comes along, whatever new thing, the stimulus that comes along in the environment, whatever distraction comes along, you're going to react to it. We can't engage in goal-directed behavior. We can't build buildings. We can't write symphonies. We can't graduate from MIT unless we can control our own thoughts and focus our thoughts on goals. Working memory is the fundamental mechanism by which you do that, by which you wrest control of your thoughts from the environment and focus your thoughts on what you want to do. And that was never addressed by the classic model of working memory. That was something that was left to, we'll figure that out later on. But the thing about these sort of observations is they provide new clues into how we control what we think about. Okay? And that's what I'm going to tell you about next. Now, one thing you may notice from this figure here I showed you is that the gamma, so far I'm talking about the gamma bursts holding the working memories, right? I haven't mentioned these beta bursts yet. The lower frequency beta bursts occurring here. One thing we notice is that the gamma and beta bursts are not occurring at the same time. So you see the gamma bursts are occurring here, 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 and the beta bursts tend to happen between the gamma bursts. And you can see that more clearly here, where now we're plotting, this is now an average across trials, so we can plot this sort of dynamics, is we're plotting the average gamma burst here on the upper line, and the beta burst here on the lower line. So here's time over the x-axis. This is about a 2-second trial. The animal's holding things in working memory. two pictures of working memory, S1 and S2. Here's the power. And what happens is that the gamma bursts go up whenever the animal's looking at the picture, because then the gamma bursts are associated with this stimuli the monkey's holding the working memory. The picture is the monkey's holding the working memory. So they go up whenever the animal's looking at the picture, and they maintain a high level over the memory delay. Now look at the beta bursts, which I haven't really talked about much yet. The beta bursts are like mirror opposite of the gamma bursts. Whenever gamma goes up, beta goes down. Gamma goes up when the animal's looking at the picture, beta goes down. In the memory delay, gamma initially drops, beta goes up, and as gamma rises, beta drops. They're like mirror images of one another. So what this suggested to us is that this could provide a mechanism for volitional control of working memory. If gamma really holds the working memories, that means that turning up and down this beta signal can turn up and down gamma, and thereby control access to working memory, control working memory storage. If I want to load something in the working memory, I turn down beta, gamma can grab whatever I want to hold in the working memory. If I want to clear out working memory, I turn up beta, gamma turns down, and my working memory is cleared out. So it occurred to us that these beta and gamma dynamics, which by the way have never been observed before, may be a way for exerting volitional control over working memory. So I talked about gamma being linked to the sensory information the animal or human is holding in working memory. What is beta doing? When I tell you what beta is doing, let me introduce you this concept of bottom-up versus top-down processing. What bottom-up processing is that's the sensory information. That's the stuff we've been talking about so far, the stimuli, the pictures the animal's holding in working memory. That's bottom-up information because that's information from the outside world, the raw information from the outside world entering the brain. You show the animal a picture, the picture's in the outside world. Because that's a sensory input, we call that bottom-up information. It's something the brain is copying from the outside world. Now, by contrast, there's something called top-down processing, and top-down processing is the knowledge you've acquired through experience. It's the things you know. It's the expectations, the knowledge. It's your knowledge of how things work. It's top-down knowledge is what's keeping you in your chairs listening to me because you know that that's what's expected of you in a lecture. It's the top-down knowledge is your brain, is the cultural knowledge your brain has absorbed for how the world works and how to navigate situations like this and many others. Okay? So gamma, as I mentioned, is associated with this bottom-up sensory information the animal's holding in working memory. Well, many experiments from our laboratory have shown when we look for how this top-down knowledge the animal has acquired, how that is expressed, it's always expressed in beta. Never in gamma, always in beta. So these two rhythms I've been talking about, they reflect the two basic modes of knowledge, of information your brain has. Sensory information in gamma versus your actual experience knowledge expressed in beta. So what this means is that beta and gamma interactions can provide a means for top-down knowledge, again expressed in beta, your experience, you knowing what to do, that can regulate the processing of bottom-up sensory information in gamma. In other words, control what you think about. So what we've done is we've tried to, this is all brand new data now, we've explored this further because we wanted to understand how these beta and gamma rhythms interact. So what we did is we used this brand new technology to record from multiple layers of the cortex simultaneously. Maybe you'll know this, but your cortex, the outer rim of your brain, isn't just one thing. It actually has 6 layers. Each of these 6 layers has unique cell types and has different connections with the rest of the brain. The important ones I'll get to in a moment. So we wanted to figure out what it is, how these two rhythms interact to control working memory. So we decided what we do is we'd use these brand new technologies, brand new probes here to record from, this is a probe here with multiple contacts by which we record electrical activity all along the shaft here. We introduce it down to layers of cortex and record from all layers of cortex simultaneously. And by the way, there's also something that people hadn't done for 50 years. They've just popped their electrodes in the brain and didn't worry about what layer of cortex they're in. But we figured if we want to look at how these rhythms interact, we need to understand how all the cortical layers interact. So, the first thing we found is that we'll separate now the layers into superficial and deep. You'll see why in a moment. Superficial layers and the middle layers above, deep layers and the middle layers and below. And the first thing we found is all this spiking that's carrying the information the animals holding a working memory, they're all occurring in the superficial layers of cortex, not in the deep layers of cortex. So why is that important? That's important because the superficial layers are the feed-forward layers of cortex. They're the layers of cortex that carry sensory information, bottom-up information from the outside world deep into your brain. So if the animal's holding pictures in working memory, animals holding information from the outside world of working memory, we'd expect that information to be in the superficial layers, in the bottom-up feed-forward layers of cortex. Okay? Now, then we looked at the rhythms. Now this is a plot here. Sorry, I should have mentioned this. This is time, and this is depth of cortex, and this is just spiking activity here shown in red. Now we're looking at different layers of where we see the beta and gamma rhythms. Remember, the gamma is the bottom-up layers, beta is the top-down. Gamma is the bottom-up rhythms, beta is the top-down rhythms. And what we find is that there's more gamma in superficial layers of cortex and more beta in deeper layers of cortex. And this makes sense because, again, gamma is associated with sensory information we have in working memory. So gammas in the superficial or feed-forward layers of cortex, they're carrying sensory information deep into the brain. Whereas beta, top-down information, they're occurring in the deep layers of cortex, and the deep layers of cortex are the layers of cortex that carry information in the opposite direction. From the front of your brain, the executive part of your brain that has all the knowledge, that's the deep layers of cortex, carry that executive knowledge from the front of your brain back to the rest of your brain. So the beta rhythms, which we have associated with top-down knowledge, are happening in deep layers of cortex, which is where we expect them to be, given what we know about the anatomy of the cortex. Now, if you look at these two rhythms, how they correlate one another, what we find is that the beta rhythms in a deep cortex, carrying the top-down knowledge, are anti-correlated with the gamma rhythms in superficial layers of cortex, which are carrying the bottom-up information. So that's shown here. Here's the different layers providing either superficial or deep, providing beta or gamma. The important thing here is that the red colors are beta, the blue colors are gamma. And this is a power correlation here. Blue is anti-correlated, red is positively correlated. So what this just means is that the beta and gamma rhythms in the deep layers versus superficial layers of cortex are anti-correlated with one another. Whenever one goes up, the other one goes down. Okay? So in other words, this supports this model in which the deep layers of cortex are carrying your knowledge from the front of the brain to the back of the brain. The superficial layers of cortex are carrying sensory information and gamma rhythms from the back of your brain deep into the front of your brain. And these two interact in this anti-correlated way, allowing the top-down information in deep layers and beta to control, regulate superficial layers of cortex, thereby regulating what you control, what you hold in working memory. So to just sum up this model is that we believe the way working memory works, the way you gain volitional control of your own thoughts, is that strong deep layer beta, the deep layers of cortex that carry feedback information from the front of your brain to the back of the brain, they carry top-down information, and they regulate the weaker beta rhythms in superficial layer cortex. That in turn regulates the superficial layer cortex gamma, which is carrying the sensory information, the feed-forward information from the outside world deep into your brain. And this in turn regulates the spiking that stores bottom-up information in working memory. In other words, this dance, this interactions between top-down information expressed in beta, in deep layers of cortex, and sensing information carried in superficial layers of cortex in opposite directions, they interact with one another in this anti-correlated way that allows you to control your own thoughts, allows you to control what you think about. Now, I can't tell you why you think the way you think. I can tell you why you have the goals you have. I can't tell you yet why you make the decisions you do. That's all stuff we're still working on. But we think this is the fundamental infrastructure by which, the platform by which your brain gains control over its own thoughts. And as far as we know, this is the first time someone has actually shown an infrastructure by which volitional control can act. So I'm going to go on to another subject now, but before I do, is there any questions on this? Yes?
So I don't understand how you represent structure and substructure. For example, if you have a coffee cup and a handle,
Yeah.
and they're different colors and so on, how can you, and depending on what you're doing, you can be interested in one part or the other, or the orientation of the cup or so on. It seems like if you're only doing that in very close terms, in terms of beta and gamma, and there's many objects that you might be interested in or not interested in. If you look at a car, depending on the level, there's many objects. I don't see how you could possibly represent it just in that very coarse way if it has two sets of brains.
Absolutely. I did not talk about that level of understanding yet, but yes, you're right. You have objects that control multiple parts. You can either choose to focus on the whole object or individual parts. There's a lot of neurons, especially in your higher parts of your visual cortex, that piece things together in this gestalt way. But neurons in the back of your brain, they pull things, they represent things more by the parts. So this top-down information can choose to act on different levels of processing your cortex that can focus on your individual parts or the whole. So that's a simple way of putting it. That's pretty much the way it works.
So in terms of neurons, what's the difference between focusing on the entire cup and focusing on the handle? Is it a different population?
Different
or
population.
is it superposition of the same thing over the same population?
Yeah,
I'm giving away my biases here. But
I love talking to MIT people. It's a little of both. So it's a level of analysis in cortex that gives you either the parts or the whole, but the whole, your perceptions can depend on the interaction between the two. It's all a matter of which one's emphasized at a given moment, and that's what the top-down information is good for, is choosing which level to emphasize in a given moment.
Could you quantify some of this, at least in order of magnitude terms? How many bytes of information are in working memory, and how long does the information persist there?
That's it. You guys are awesome. That's a great question. It's not easy to answer because your brain does something called chunking. Okay? So you can represent things as individual parts or chunk them together into one unit. And something I was going to talk about in a little bit, but so your brain is very good at data compression. I mentioned that you have a very limited capacity to hold, the average adult human can hold 4 thoughts in mind simultaneously. How does your brain deal with that? I mean, it's a severe limitation in bandwidth, right? The way the brain deals with that is a process called chunking, or we can call it categorization. You have a word like peace or justice. That's one word that stands for a lot of different things. So having this umbrella by which we can add labels of things and chunk them is the ultimate form of data compression. So it all depends how many bits. It all depends, it all depends on what level we're talking about. Okay, so that's a difficult question, but the brain can represent things as gestalt wholes of chunks rather than each individual detail.
No, but you know how many neurons there are and what each neuron can hold. I mean, order of magnitude.
No, you're assuming more knowledge than we actually have. The brain's very, very complex. We know that ensembles represent memories, right, or thoughts or perceptions. The fundamental unit of the brain is a collection, a network of neurons that represent thought. Well, we still know exactly what, and we know the ensemble should exist. We still know how ensembles are formed. I have some ideas of that. That's a whole other different talk. But it's not a matter of knowing how much information each neuron can represent, because each neuron is embedded in this ensemble, and it's the ensembles that represent information. And that's going to be a highly dynamic process where ensembles could swell or get smaller or dynamically change depending on the demands of the task. So the answer to that question is not as straightforward as you think it is.
And you looked at non-working memory, so basically, I mean, I know when I go down to the basement, I can't remember why I went down, so I have to go back up and get the same cue and everything else. So, you know, I'm sure that they have, you know, models of animal models that will have dementia or some sort of thing like that. And
Sure.
have you been able to correlate any of this to what they don't have? What's different about those animals versus the regular models?
Sure, that's an active area investigation. I don't study everything in my lab, but that's one of the most interesting things about the brain is that we have the ability to store a lifetime of information in our heads, right? I mean, in latent form, in long-term memories. But only can think about a few thoughts simultaneously. That's a really fundamental mystery in your brain. As far as we can tell, these kind of memory lapses that you're talking about are not so much a storage failure or the memories are lost, you just have trouble retrieving them. They're still there, you just have trouble accessing them because the links get lost. Now, how that process happens, I wish I knew.
Yeah, you do know.
I'd have a Nobel Prize if I knew the answer to that.
Right.
This question has to do with sort of the evolution of this concept. I remember distinctly lectures given by Jerry Lettvin back in 1968, where he talked about a feedback mechanism in terms of the function of the brain and the personality, with the feed-forward path being sensory data that was being modulated by the feedback path. He didn't have the deep layers and superficial layers, but I think he had some of the rest of it. Did this come from that, or is this sort of a parallel evolution?
Partially, yes, it did come from that. I mean, like, I've met Jerry lots of times, and he was a real character. A lot of the concepts we have now, actually, you know, as my PhD advisor used to say, there's no new ideas, just old ideas rediscovered. And Jerry had a lot of these really he had a lot of these really avant-garde ideas that proved to be true in the end. He thought deeply about how the brain works, and he thought about this stuff and got a lot of things right without having any of the knowledge that we have now. He just in his own thoughts. So he was an amazing guy. He didn't have the superficial deep layers things. He was studying animals out of cortex, but he definitely got the balance right. That brain processing is not just feeding stuff into the brain. It's an interaction between the feed forward and feedback. So yeah, Jerry, inspiring guy. Yeah.
Can you talk about what happens physiologically to generate the gamma vibrations and the beta? Is it a circuit of neurons that just kind of goes around a bunch of times and that creates kind of this vibration that you can detect?
It is. What it is a balance between excitatory and inhibitory neurons in the brain. So excitatory neurons are recurrent connections trying to excite each other. Then they interact with inhibitory neurons trying to shut each other down. And it's the balance between them. How much excitation and inhibition you get that either causes the gamma rhythms or the beta rhythms. It depends on which ones are essentially winning out at different levels.
So why are the betas lower frequency? Is it because there's more neurons involved with that? It takes longer to go around the circuits?
Good question. Don't know the answer. I can say from a functional standpoint, if you're going to have top-down information expressed in the brain, a signal, you want to have lower frequencies. These lower frequencies are more of a catch-all basket. Higher frequencies are less forgiving of when spiking is actually occurring. The low frequencies can grab spikes into their well, if you will. So low frequency rhythms can push around high frequencies more easily than the other way around. And you want a brain where the top-down stuff is pushing around the bottom-up stuff. You don't want a brain where the bottom-up stuff is pushing things around, otherwise you have Phineas gauge. So it makes sense from a functional standpoint why you'd have the lower frequencies being the top-down, the higher frequencies being the sensory information. I do have a whole other part of my talk, so I'll save a few questions at the end. Hey, Rusty.
Hi. So we know fairly well about how individual neurons impact other individual neurons. You've given us a little story about how the interaction among neurons creates the gamma and beta rhythms.
Yep.
Question is, mechanistically, how do we know how the beta shows off the gamma or vice versa? Is there an actual causal relationship, or is this essentially switching between modes? It's not causal.
Well, we believe it's switching between modes and causal. And we're trying to do experiments now where we're actually changing the beta rhythms, strengthening them, increasing them, and seeing what effect it has on the gamma rhythms. Not just watching them work.
Yep.
We actually manipulating them, and we do see exactly what we expect. When we increase the beta rhythms, gamma rhythms go down. When we relax the beta rhythms, gamma rhythms goes up. We're doing that doing new optogenetic experiments, which I know you're familiar with, Rusty, in the brains of animals. So, it's important to have these cause and effect relationships, and the cause and effect relationship does seem to be there between beta and gamma.
So if you're sticking a probe onto a single neuron, and then you're showing an animal a picture of flowers or something, how do you have any confidence that that one neuron cares about flowers at all?
Great question. So the way we know the neuron cares about is we show the animal a picture of flowers and a bunch of other things. Flowers, house, tree, a dog, a cat, whatever. And what typically happens is the neuron will give off spikes, whereas we show the picture of the flower and doesn't give off spikes, it gives off fewer spikes to other pictures. So we look at we get information by changes in the spiking rate of neurons to different events, different pictures, et cetera, and so forth. We actually compute information from that. So that's how we do that.
I have a physical question.
Physical questions?
The slide at the beginning of the presentation showed the picture of a neuron.
Yeah.
And the neuron has, I don't know, 15 different arms radiating out from it. And we're talking here about one wave interacting with another wave. What happens in that neuron when there's 15 different connections going to other places? Is it doing 15 different things or one thing? Or what is the connection between the physical geometry of a neuron and how it interacts with others?
Such an awesome question. So, for 50 years of systems neuroscience, it was thought that anatomy was destiny. If two neurons are connected, if a neuron has 15 connections, it's connected to 15 or more, actually neurons have thousands of connections, it can do 1,000 different things, right? Or you said signals 1,000 different ways. Our studies of the role of brain waves has led to a very different view of how the brain actually works. Think of this as if you have two sets of neurons, two different parts of the brain, if they oscillate together at the same frequency in sequence, they're going from a state where they're relatively low energy, high energy, simultaneously, right? That means they can communicate one another. They're both in active states, so one can, when one's giving off pulses, spikes, the other one's ready to receive it because they're both in an active state. When they're quiet, they're not talking to one another. Now imagine a situation where two sets of neurons are firing, they're oscillating out of phase of one another. That's a way of preventing communication, because when one's giving off spikes, the other one's not in an energy state to receive it. So our lab and others have revived this idea that anatomy is destiny. We don't think of so again, the way it was thought of is that if neurons are connected, they're going to talk to one another. The way we think about it now, as a result of this kind of work and other work, is that anatomy is like the road and highway system. It says where traffic could go. Where traffic actually does go from moment to moment depends on these patterns of resonance. Neurons synchronizing their waves across different parts of the cortex. They're essentially the traffic lights that determines which neurons talk to one another. So the answer is that if they have this neuron giving off 15 or 15,000 different projections to other neurons, not all of them are going to be working at a given moment. Which ones are going to be working depending on which patterns of resonance the neurons are resonating with.
Could you use the phase relationship of alpha-gamma as a diagnostic tool for autism?
Great question. We think a lot of neuropsychiatric disorders may result from these mismanaged rhythms.
Yeah.
So you can imagine, like, if the beta rhythms are weak, you're going to have a brain that's too sensitive to sensory input. That's a lot of what autistic brain does. It can't filter out all the raw sensory inputs. We have other data that's showing that is juggling the act of different rhythms, juggling different working memories. They also play a role in ordering the memories based on these phase relationships. So you can imagine if that gets disrupted, you have disordered thought, like in schizophrenia. So we do think a lot of neuropsychiatric disorders can be explained by dysfunctions in these rhythms.
if I can't interpret your expression at the right time, see, then I'm autistic.
Yeah, exactly.
So the input and output in different directions.
Yep. Oh no, I think your insight is absolutely right. And you know, there's evidence even on the behavioral level that when you're paying attention, when you're perceiving, you don't actually perceive in a steady state. You're sampling the world in this rhythmic way.
Yeah.
So disrupting those rhythms can disrupt a lot of things. Yeah. So I want to go to
we're not on the Q&A yet, right? Yeah.
Now let's start. So I want to switch gears just a little bit and talk about. So I've been a basic scientist for many years now. And one thing about basic neuroscience, basic science in general, is that occasionally something practical comes out of it. In fact, there's you hear translational research, it's clinical research, right? It's like, how are we going to combat this disease in the next few years? I think of basic science, like what I do, as like clinical science, only with a much longer window. We discover basic truths, so eventually something practical can come out of it. Well, something practical came out of this work, my work on working memory. I thought I'd tell you a little bit about that so you could see how we could take some basic discovery and actually turn it into something. So I'll switch gears for a moment, and I'm going to talk about something I've been mentioning, and that working memory capacity. We have a very limited capacity to take in information. Working memory is the sketchpad of your consciousness. If it's limited capacity, you're limited in how much information you can actually perceive, process, be conscious of. As a result of this, you don't always see what's in front of you, and we discovered something that could help mitigate that. So, do you think you see everything in front of you right now? Right now, you probably have this perception because your brain is giving you this perception. By the way, your brain is really good at deluding itself. Right now, it's deluding itself into thinking that your mind is like is a video camera with a wide-angle lens, and everything in front of you you're just taking in, right? That's an illusion. Your brain is actually sampling the world in little bits and pieces and piecing it together into an illusion of seamless perception. Don't believe me? Let me prove it to you. I'm about to show you a picture. It's going to flip back and forth between two different pictures. And something can be different between two pictures. I want you to raise your hand when you knows what the difference is. And Rusty, you're not allowed to respond to this. So ready? Raise your hand when you see the difference between the pictures. Okay, some of you are starting to get it, so it takes a while, doesn't it? Another little detail, the shadow here under the helicopter is appearing in this area. This is a little tiny feature. It's a major thing in this picture, right? Here's one. Ready for another one? Here's another one. Okay, so you getting it?
Oh, yeah.
The tree here is appearing and disappearing. One more, just to, as if I haven't proven this point already. These leaves are appearing and disappearing. Right? So what's going on here is your brain isn't perceiving everything that's in front of you. What your brain is doing is anticipating which parts of the scene are important, focusing on them, like the house here and this and consider everything else to be background, basically filling in the blanks. That's what your brain is doing. That's the way your perception actually works. Think about this next time you're driving in a car. So, this is something called working memory capacity. And again, I referred to this earlier. You actually have a limited capacity to think simultaneous thoughts. Your average adult human can perceive about 4 things, perceive and hold in working memory about 4 things simultaneously. But it varies highly from person to person. Some people can do 2, some people can do 8. Interestingly, this correlates highly with measures of intelligence. So here's a plot here of a new test that the NIH developed to look at something called fluid intelligence, how well, in a non-culturally biased way, how well you can make judgments about fluid intelligence. That's shown on the y-axis here. On the x-axis is the age-adjusted storage capacity of a bunch of different subjects. And you see that the R squared here is 0.46, meaning that we can explain 50% of the variance among people in measures of intelligence just by knowing how many pictures they can hold in mind simultaneously.
Is there a gender difference?
Not as far as we can tell.
Is that what?
Gender, is there a gender difference?
What does that mean, hold in mind? To what level of detail or lack?
I'll show you an example of that in a moment. It's a simple test. I show you a scene, I show you another scene, something's changed, tell me what's changed. The question is how many things are there. It's a very simple test, but it captures, in a big way, measures, it's highly correlated with measures of fluid intelligence. Now, here's the thing. So I said your average adult human could hold 4 pictures in mind, and I'll show you the test in a moment, 4 pictures in mind simultaneously. This actually changes as you age. So when you're young, again, it varies highly from person to person, but the average is 4. When you're young, it starts at about 2. When you get to be a full adult, it goes up to about 4. And then when you get to be older, it drops back down to childlike age. However, and I'm not just saying this because I'm rapidly approaching this level right here. I mean, I have no idea. I'm really on the cusp right there. You know, your cognitive bandwidth, the amount of stuff you can hold in mind simultaneously, drops down to childlike levels. It shrinks when you get older. However, an older brain has an advantage that mitigates this. We have wisdom. The ultimate form, I'm not making this up because I'm getting older. I swear. Wisdom is the ultimate form of data compression in your brain. You have knowledge like peace, love, justice. Those are single words that stand for so much. And they stand for so much only with experience. This is how your brain learned to deal with this limited capacity of working memory. And learn to compress data by compressing things into under labels, giving you wisdom. So you may get, your capacity may drop when you get older, but you're much wiser and you can deal with it better. In fact, you can deal with it better than a young person with higher capacity. And maybe that's just me talking. But we all agree, right?
Yeah, absolutely.
Now, what's interesting about this capacity is that, first of all, it is temporarily reduced. So so if I know what your capacity is, let's say it's 3, let's say it's 4, let's say it's 5, 6, 7, 8. You're a bunch of MIT graduates. I'm guessing your guys are all like 7 and 8. You're way up here on the spectrum here. Sorry, the spectrum, intelligence spectrum. What's interesting is that this actually reflects your very stable over years. So if you have a capacity one day, if I test you a month from now, 2 months from now, 6 months, a year from now, you'll be at the same capacity. Yeah.
Can you train for it? If you have somebody who does it, multiple times, like the image, you showed us, and then you cannot stop looking at the difference, right?
I'm sorry to say you can't. Your capacity really doesn't change as a result of the training. That's what luminosity. You guys know luminosity? Yeah. That's what luminosity tries to do. Little sidebar, luminosity does not work. Don't waste your money. If you practice luminosity, you will get better at the luminosity test. There's exactly 0 evidence it actually translates into the real world. So don't waste your money. But that's one thing luminosity tries to do. And it'll give you a working memory test, and you will appear to get better at it, but what you're doing is just finding better strategies for chunking the information, not really increasing working memory capacity. So, your capacity is very stable over years, but it can drop temporarily if anything disrupts your cognitive function. Let's say you're sleep deprived. Let's say you're stressed. Let's say you had a fight with your spouse. Let's say you're worried about something. Something's going to impact your ability to do your job, your cognition. There'll be a temporary reduction in working memory capacity. And also there's a permanent decrease in working memory capacity with diseases that impact cognition. Dementia, Alzheimer's, schizophrenia, Parkinson's, anything any disease that produces a cognitive impairment will be reflected in a decrease. Yeah.
What causes a temporary reduction? Is it less electrical voltage inside the brain, or what?
Well, what it is that there's a certain level of hormones in your brain. So stress produces a release of certain hormones in your brain, and there's a certain, all of biology is a converted U curve. There's a certain range of things that work really well. But what stress can do is release too much of these stress hormones and knocks you out of the optimal range, and that reduces the information processing in your brain, by which mechanisms we don't really quite understand yet. But you can measure the reduction in information processing when you get knocked out of that range, when you have too much stress and these hormones get released. So it's a very chemical, physiological process. So.
Or you could imagine if you're worried about something that's taking up a slot.
Yeah, and that's another way. There, just simply worrying about something else is taking up space in the limited capacity of working memory. Exactly. But it's also more general in the sense of just stress in general will reduce it. Yeah.
It's, you haven't got to it yet, but it's a sort of age-related phenomenon, the efficiency of transfer from short-term to long-term memory.
Efficient transfer, age-related.
Age-related.
It's a good question. I don't know the answer to that, actually.
The efficiency of that transfer process will degrade quite effectively.
Yeah. I mean, you retrieve information.
You know it, and then you don't remember it.
Yeah.
And you really have to rehearse it over and over again if you really want to get it into long-term memory.
Yeah, that's a great question. I don't know the exact answer to it. I'm guessing it's more of a the more stuff you have in long-term memory, the more links there are, the more possibilities.
Yeah, but you worry about it later.
Yeah. I'm worrying about it now, brother. So, let me show you how. So, I've showed you this. We test working memory capacity typically using visual tests. And the way we test it is very simple. So, here's a test where what you do with the subject is you have the subject stare at a spot of light. You present a scene of like 2, 3, 4 colored squares. They go away for a second or so, then they come back. One of the squares has changed colors. Tell me which squares change colors, or let's tell me whether any of them has changed colors, right? And if you're below capacity, let's say you have a capacity of 4 or 5, if I use 2 or 3 colored squares, you'll get it right every single time. But if I exceed your capacity, you're going to get it wrong. You'll get it right occasionally, because occasionally one of the things you hold makes it through will be the right one. But you'll make some errors because occasionally you're missing some things, right? So people have been studying, even predating Hubel Wiesel, even predating modern neuroscience. People have been studying working memory capacity for 70 years or so, since George Miller, no relation, in the 50s, first started studying working memory. Now, in 2011, our lab made a remarkable discovery. We didn't anticipate this, we didn't expect it, but it came out of data. We found that you have actually independent capacities on the right and left sides of your vision. In other words, you're looking straight forward here, you have a capacity over here that's independent of capacity over here. So what that means is you don't have a capacity of 4, your average adult doesn't have a capacity of 4, you have a capacity of 2 on the right and 2 on the left, as if you have 2 brains in your head. One brain looking at the right side of vision, one brain looking at the left side of vision.
Is there any correlation between right and left brain?
No, there's not actually. I can give you, I have a whole undergraduate lecture I give at MIT about right versus left brain. The thing about parenthetically, the thing about right versus left brain is a lot of those claims are great. There's a kernel of truth to them. They've been greatly exaggerated. But there's no relationship between right and left brain type personalities and the capacity of right and left. So we first discovered this independence of right and left vision. We went to a meeting, and there's a whole meetings about working memory capacity. Whole meetings about working memory capacity. And we presented this, and people in the audience were like, no way can this be true. This is true. We discovered it 40, 30, 50 years ago, right? We said it for 50 years. And I said, have you ever really looked for it? And the answer was no, no one has. So since then, people went back to the meeting, went to their own laboratories and looked at their own data, and it turns out that we've now been replicated a dozen times now. So what was first, this can't be true, turns out to be absolutely true. So this is the data here, this is data from our laboratory. What this shows here is, let's say the subject doesn't know which of these squares are going to change color. It's random. But the one that's going to change color we'll call the target. And for the sake of plotting here, we've always had the target here on the right side of vision, other things on the left side of vision. And what the color shows is how well the subject performs. So you look over here, here's the 2 squares. T, the target's going to change color, this one's not going to change color. The performance is nearly 100%. You add stuff on the opposite side from the target, it doesn't change the performance at all. It's still 100%. Now look down the column here where we're adding stuff on the same side of the target, and performance just drops dramatically. And we don't just see this on the behavioral level, you can see on the neurophysiological level. Whatever happens on the right has no effect on information processing on the left, and vice versa. I mean, remarkable. When my student, Jim Bushman, discovered this, I said, this can't be right. Something must be wrong in your code. Go back and check your code. Well, we checked it, wrote brand new code. It's true. So what does this mean? Oh, one thing I'll point out, this is a right-left side of your brain effect. It's not a right-left eye effect. Here's the way the anatomy of your brain works. You have your two eyes here. The left side of your vision goes to one side of your retina. The right side of the vision goes to the other side of the retina. Then they cross in the optic chiasm, and all the stuff on the left side of each eye goes to the left brain. All the stuff on the right side of each eye goes to the right brain. You can get the same effect, this whole independence, by just covering even with one eye. So it's not an eye thing, it's a brain thing. It's a brain hemisphere thing. So what does this mean? This means that people on average don't have a capacity of 4, they have a capacity of 2 on the right and 2 on the left. Now right here we've learned something. Let's say you have a heads-up, every car manufacturer, jet pilots, airline manufacturer now working heads-up displays where information is presented on the windshield you're looking through. If the display has 3 important things, you want 2 on 2. You don't want 3 on. You don't want 2 on the right and 1 on the. You want to balance out the display so you're using each capacity. In other words, if the display has 3 important things, you want one to be on the opposite side, so you don't overload the capacity on one side because you'll miss something. Also, every little thing uses a bit of capacity. It's not like you have slots in your brain. Every time you process something, it's degrading information a little bit. So if you're monitoring 2 important sources of information, you want them on the opposite sides of vision so you can devote full resources of each. But as it says here, wait, there's more. It turns out that different people have different capacities on the right. Different people have different capacities. Okay. Now, when we realized that some people have a capacity of 2, some people have a capacity of 8. The average is 4. We thought, well, if the capacities on the right and left are independent of one another, why should they be the same? Why should it always be 2 and 2? So we did testing in human subjects. Turns out it's not true. Different people have different capacities on the right and left. Some people have a capacity of 1 and 3, 3 and 1, 2 and 2, and so on. And because different people have overall different capacities, there's a lot of variation on the right and left side between different people. Some people have 5 and 2, 1 and 5, 2 and 2, 1 and 5. So in testing this further, even more, we found that although up and down is not a bias, there's also an up and down effect. So up and down is not independent, only right and left are. But there's even a bias on up and down. So people have different capacities, different quadrants of their visual field. So again, I'm looking straight ahead. I have different capacity here, 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 and here. It's not a compartment thing, it's more of it's an even spread. But people have hot spots where they have high capacity, and cool spots where they have low capacity. And this varies highly from person to person. Yeah?
I seem to remember reading that when you remember something, from way back, like you look top right or something like that, is this related to that fact, or is that a fact at all?
It could be, because we started testing subjects on our app, which we use to measure this. People would say, like I found somebody who, one subject said, oh yeah, they have a much higher capacity on the left than right. They told me, when I was in college, I always sat on the right side of the classroom, so again, all this lecture was on the left side mainly. And whenever I sat, whenever the seat was taken, I sat on the other side, I felt I wasn't getting as much out of the lecture. Me, I have a left side bias. And for years, before I knew this, I have 2 computer screens. I have the screen in front of me, and I have a second screen. I've always set up so there's one screen in front of me and one on the left. Now, why not on the right? Why left? I don't know, but apparently my brain realized that I have this left bias and put the information in the hot spot of my vision. Right? So, different people have different perceptual capacity, different capacity to take in information in different parts of their visual field. Yeah?
I was wondering how tension factors into this talk about a bias, could it be a bias of attention rather than a bias of memory?
It's a perceptual bias, so it's information reaching the conscious parts of your brain. But you're absolutely right. Attention is a big factor in that. So, for example, I'm skipping ahead a bit here, but if you know your capacity is uneven, you can take our app and train yourself and actually spread out your capacity. So you can go from a capacity where you have like 1, 4, 4, 4, and learn to spread out your capacity more evenly. And what you're learning to do is you're learning to allocate your attention in a more effective way that makes up for the biases. It's a great question. So you want to know how different people are? Maybe these are all just. I mean, it's actually more of a continuum than these boxes I showed here. Maybe I'm just making these numbers up. Well, I'm not making these numbers up. So here's a test of 220 humans. And what the scatter plot shows is how much bias these people have in different parts of their visual quadrants. So each dot is a different subject. If each subject had no bias whatsoever, their dot would be right in the center of the screen here. But as you move out, these subjects have more and more of a bias. And look how much bias you see. Very few people are in the center of the screen. Very few people are even in different parts of your visual field. Half the subjects had a bias of at least 25%. They're 25% better in one direction or another. And a good number of subjects are 50 or 75% better. They're nearly twice as good in one part of their visual field than the other.
Is this all within Western culture?
Great question. One of the we have a company which has an app which measures this, and one of the members of our advisory board has a satellite lab in China, and we're very interested. Is this all Western culture bias? Maybe more biased towards the left because we read left to right. It doesn't turn out to be true. You have Chinese citizens who read up and down, same biases. Doesn't make a difference. Israeli subjects who read opposite doesn't make a difference. It seems to be something very innate about your brain that's not affected very much about culture. So these effects are huge. Sorry?
Languages are only 2,000 years old. That's not enough time to tell much.
Yeah, that's right. That's right. Brains take a long time to evolve. Totally. So, these effects are huge. These biases are huge, huge. And again, I don't think it's any accident that for years I set up with my second computer screen on my left side where I know now I have a huge bias. So I'm telling you about this because, we do basic research and occasionally something comes out of it that we think, wow, we can actually do something with this. And I'm excited by this because I think making a practical application of basic science is a great way of showing that your basic science is working. There's nothing more, no better test of basic science than to put to practical use. So what can we do with this? Well, we have a company which we formed called Split Sage, where we actually have an app which gives you an eye exam for the brain, where we identify blind spots where you have low capacity, sweet spots where you have high capacity. You take our test, it only takes a few minutes, and you get a heat map of where you have high capacity and low capacity. So what's this good for? Well, this match is good for a lot of things. Anything that involves split-second decisions in performance. Athletics. You have a quarterback you paid $65 million for. Don't you want to know where that quarterback has a bias at the left, right, or up and down so you can learn to compensate for it? Military, police, you're dealing with split-second decisions in difficult situations where you're taking a lot of information simultaneously. Knowing where your biases are can help you mitigate those biases. And in fact, we know you can mitigate the biases because if you train subjects on our app and allow them to give them feedback, they can actually even out their biases. Putting teams together, let's go back to the football analogy. If you have a team of football players, some are strong on the right, some are strong on the left. You want to line them up in the right way, so you're speaking to you're ordering them towards their strengths and lining up away from their weaknesses, right? Transportation, heads-up displays for airline pilots. Amtrak employees, all these different manufacturers for airlines, for cars, for trains are developing heads-up displays. Now, with our app, what you can do is you have a heads-up display for your car, looking through your windshield. Well, it's going to give you critical information. Well, I don't want to overload you so you don't see what's on the road in front of you. You can take our app and you can design your heads-up display for your car. Again, the app only takes a few minutes of testing. You got to do it inside the car. And now you have a personalized heads-up display that gives you the information you need without overloading you so you don't miss what's on the road in front of you. If we can reduce car accidents by 5%, that's a huge number of lives saved. Now, other applications other than this performance application is that, remember I mentioned that this cognitive capacity, working memory capacity, is temporarily reduced by sleep deprivation, anything that's going to impact your ability to perform your job is going to result in a decrease in your working memory capacity. Well, what we can do is we can use this as like a breathalyzer for your brain. We have a baseline heat map of you, knowing what your capacity is, different parts of your visual field, knowing what you are when you're functioning normally. But now you're an airline pilot, you come in every day, you take our test for a few minutes, and all of a sudden every day, day in, day out, your map looks normal. Then one day you come in, and the heat map looks cooler. Now all of a sudden your capacity has dropped. And incidentally, it's a sensitive measure because your capacity drops first where you have the highest capacity. So knowing you have a high capacity, say in the upper right, that's going to show the most effect. So it's a very sensitive test of when you are off your game. So you come in before the airline pilot, for the Amtrak engineer, gets into that critical job. They take our test. Oh, your map's a little off today. Maybe somebody should be talking to you. Maybe you shouldn't be doing your job. Maybe you're sleep deprived. Maybe you're stressed. Maybe something's preventing you from doing a job that other people's lives depend on.
Maybe you're on the cell phone.
What's it?
Maybe you're on the cell phone.
Yeah, well, you have to put your cell phone away when you take our test. And hopefully they're doing that when they're flying the plane or
driving the train.
Driving the train. Yeah, I know that was an issue with this one train crash, but that's a whole separate issue. And finally, as I mentioned, it's very stable. This working memory capacity is stable over years and years and years and years. You test people 2, 5, 10 years, and their working memory capacity stays about the same, just like their cognitive function, their intelligence level stays the same. But with some diseases like dementia, Alzheimer's disease, your working memory capacity begins to drop. And it will begin to drop before you start showing real strong behavioral manifestations of that disease. Our app can actually test when you begin to slide into that disease state, so you can have interventions earlier than later. And we all know from combating disease, early intervention is also critical. So, I think that's it. I want to thank the people in my laboratory who do all the hard work while I get to come and talk to great people like you. And I'm happy to take more questions.
Thank you very much. Like courses 902, 911, 912. There's many courses that we could follow here because I think they're pretty exciting. So maybe,
yeah, I used to teach the cognitive growth class 910, undergraduate class. Now I teach at the graduate level, so it's 901.
So I think we already asked many questions tonight, but we can stay some time for maybe like a final comment. Maybe a couple more questions. We should be wrapping up by 9. So, any other question or just be here?
Meditation allow one to flow from relationship between the beta and gamma.
That's a great question. The answer to the beta gamma thing is kind of new. I would predict that because meditation is an exercise in focusing your mind. And focus is all about top down, all about controlling what your mind is doing. So I predict that's exactly what should happen. That's a great experiment that we need to do.
And are there different bands of other than beta and gamma in your brain?
There are. There's also alpha, which is associated with unfocusing your attention. And there's delta, which is associated with sleep, and also anesthesia.
Well, there's a certain type of meditation they claim it's a theta state.
And theta, which is even lower.
Yeah, theta. That's usually transcendental meditation, which is kind of not a focused meditation. They claim to put your brain into a theta.
Yeah, and those lower rhythms are associated with relaxation and sleep. And whenever you meditate, you're putting your brain into this relaxed state, so that's consistent.
Thank you.
Thank you.